talk is by Dr. IL17 Prasad, who is Associate Professor, Department of Medical Oncology, AIIMS, New Delhi. His area of interest includes Wnt signaling pathways in cancer metabolism, cancer cell migration, metastasis, cancer therapeutics. He's done his PhD from School of Medical and Paramedical Health Science, GTSIP, New Delhi. He's won the Young Scientist Award in the area of cell biology and pathology from Society for Histochemistry Europe. Did his postdoctoral fellowship in Sweden and now working as an associate professor in Department of Medical Oncology, AIIMS. Dr. IL17 Prasad. And can I request that please stick to the time because we are a little late. Thank you.
Hello. Yeah. Good morning, everyone. First, I would like to thank the organizer of IL17 2022 by giving us this platform to discuss about our science that we are doing in Medical Oncology Department. So, again, I'm going to talk about breast cancer, but specifically on triple-negative breast cancer, which are really hard to treat. So, as already told by two previous speakers, that breast cancer is the most common cancer in India. And if you look in India compared to West, the ER negativity is high. So if you look at triple negative breast cancer cases, which are basically estrogen, progesterone, and HER2 negative, in West it is around 15%, but in India it's like 25 to 35%. And as already discussed by the earlier speakers, the breast cancer as such disease is highly heterogeneous. As you can look at here, it can be subdivided into molecular subtypes: luminal A, B, HER2 enriched, basal-like, normal-like, and T and BC falls here in basal-like. Furthermore, in 2017, they did more in T and BCs, and now we have like 6 more subtypes in triple-negative breast cancer. We call them basal-like 2, luminal androgen receptor. Then there is like MSL, which is mesenchymal stem-like, IM is immunomodulatory, M is metastatic, and then BL-like. So there are different pathways which are basically governing these subtypes. And of course, it's a heterogeneous disease. You might have like MSL and M subtype together, so how to target it? And of course, prognosis is different for each of the subtype. So there are major challenges. As already told by Professor Kundu, that it is, especially T and BC is basically occurs in younger women, less than 40 years. It is highly aggressive than other subtypes, and it is characterized by high relapse and overall survival. It reduces the overall survival. And then 50% patients which turn up to the clinic having T and BC of stage 1 and 3, almost 60 to 70% will go to recurrence. So major treatment challenges is we don't have a proven oncogenic drivers for T and BCs. Like in luminal cancer, you have ERPR, so you have hormone therapy for them. For HER2 enriched, you have like Herceptin. For treatment of T and BC, we are mainly relying on chemotherapy. Now, I did my PhD in the area of breast cancer in Wnt signaling, and we were looking like basically in triple negative breast cancer cases, this Wnt beta-catenin signaling is upregulated. So while my postdoc in Sweden, I was working again on breast cancer, but on the non-canonical pathways, and we found that these non-canonical pathways are actually down-regulated. So canonical is up, non-canonical are down, and basically this is what we think is happening wrong in triple-negative breast cancer cases. So in 2017, we published this paper in Oncotargets, and we show in this paper that aerobic glycolysis play a very important role in triple-negative breast cancers. You all know now what aerobic glycolysis is. Basically, these cells, TNBC cells, like most of the solid cancers, they actually consume a lot of glucose, and they actually convert pyruvate to lactate. the pyruvate will not go into the Krebs cycle. And this lactate is actually a oncometabolite, and it is actually transforming the cells. So in this paper, we found that there is an enzyme, you all know that phosphofructokinase 1, which is a rate-limiting enzyme of glycolysis. An isoform of it, that is PFKP, is overexpressed in breast cancer cells, but not the other two, L and M isoform. And this PFKP basically is driving the aerobic glycolysis in TNBC cells. And we also found that the beta-catenin is upstream of this PFKP in the TNBC cells. So when I joined AIMS, so we looked at this PFKP protein expression in patients. So we looked in overall breast cancer, and what we found that the high level of PFKP protein expression is associated with lymph node metastasis, and the PFKP expression is high in ER and PR negative breast cancer patients. We further validated this data in silico. On the left hand side, you can see that we have looked at the PFKP transcript profiles using Xena Functional Genomics Explorer. Basically, we compared ER positive, ER negative cell lines, and we found that PFKP transcripts are higher in ER negative cells. On the right hand side, basically we look into like online metastatic online database, and we found that PFKP protein expression in TNBC cases are associated with bad prognosis. So we know that PFKP is high in ER/PR negative or triple negative cases, and it is driving aerobic glycolysis. So how to inhibit it? So first what we did, we took this 231, these are TNBC cells, we inhibited the PFKP using siRNA, and we found that the important enzyme LDHA, which actually converts pyruvate to lactate, is also going down, affecting the extracellular lactate, as you can see in the graph. Now, till siRNA it's fine, but we have to remember that this PFKP isoform actually represents phosphofructokinase 1, which is an important metabolic enzyme and is there in the normal cells also. So how to inhibit it? And then we looked at the literature, we were finding like, can we have a natural product that can inhibit it with less toxicity? So we found Q-certin. It's a flavonoid, I will come to it. So we found that Q-certin inhibits beta-catenin in TNBCs. So we found that PFKP is regulated by beta-catenin in TNBC cells. It has also been shown in adipocytes that Q-certin can inhibit PFKP transcripts. And furthermore, in breast cancer, again, it has been shown that Q-certin can inhibit LDHA protein. So we checked with Q-certin whether it can actually inhibit PFKP. So just to tell about what Q-certin is, it's a flavonoid coming from fruits, vegetable and tea. Mainly, you can get Q-certin in large amount from onions and red apples. Q-certin has been known to be anti-cancerous, anti-inflammatory. So when we treated 231 TNBC cells, there is a downregulation of PFKP at the protein level. You can see at the immunofluorescence level, the green color, that is FITC, which is actually saying about PFKP protein. In treated cells, it's going down. In the lower panel, you can see that when we treated with Q-certin, actually, the LDHA expression also went down. And when we looked at the extracellular lactate levels, just like siRNA of PFKP, Q-certin can do everything. So basically, it is inhibiting PFKP LDHA axis and reducing the lactate. So what does it mean when the lactate is reduced from the microenvironment? Basically, lactate drives migration. And we checked then migration, and we found that 231 cells treated with Q-certin showing less migration compared to control cells. Moreover, the colony formation ability of these 231 cells was also inhibited. So overall, what we say here is that Q-certin is inhibiting PFKP LDHA axis, thereby inhibiting the extracellular lactate, which is actually driving the migration and the chronogenic potential of TNBC cells. The other protein that we are looking in our lab is RNA binding protein 2, that is human antigen R. It's an RNA binding protein, and it has been shown to have a role in cancer development and progression. So if we look at on the left hand side, actually, if you look at the protein structure, it has 3 RNA recognition motifs, and through this, it binds to its target mRNA at the 3' UTR, recognizing the ARE elements. We call as AU repeat elements. And by this, Hur stabilizes its target mRNA. This whole complex comes out of this nucleus in the cytoplasm, and basically it stabilizes these mRNA, thereby turning the protein translation of these proteins. So most of these mRNA targets are actually oncogenes, like HIF, VEGF-A, estrogen, c-MYC, which are regulated by Hur. And Hur now have been reported to actually regulate 3,500 mRNA targets. Most of them are oncoprotein. So in this figure, if the Hur is in cytoplasm, we call it as active. If it is in nuclei, we don't know the function still, so we only consider the Hur which is there in the cytoplasm. So what we looked into, we published that Hur expression is also associated with the bad prognosis in breast cancer patient, as showed in left, and Q13 can inhibit Hur by deterring the interaction with its target mRNA. So we look at the Hur expression. We published this paper recently in Nutrition and Cancer, and we show here Hur is mainly localized in the nuclei, but when we treat it with Q13, actually the cytoplasmic localization goes away. So on the right hand side, we have shown in the graphical format that it almost reduces, Q13 reduces the cytoplasmic Hur to about 60 to 80% into breast cancer cell lines, MDM231 and 468. We also showed that Q13 has ability to inhibit the migration of breast cancer cells, both the cell lines 231 and 468, and through transfer assay, and we further showed that actually this inhibition of migration is because Q13 is impairing the adhesion of TNBC cells. Then we validated these experiments by using siRNA approach, where we just knocked down Hur and showed that yes, adhesion and migration is affected, thereby saying that Q13 is actually inhibiting migration invasion through Hur. We further showed that the two downstream target of Hur, beta-catenin and CD44, were also reduced when we treated the cells with Q13. And we also explained that actually Q13 is inhibiting Hur, thereby inhibiting beta-catenin in TNBC cells, but Q13 mediated inhibition of CD44 is not dependent on Hur and beta-catenin, and might be there is an independent pathway. Now, in my lab, we are working on natural products. Basically, it's our in-house projects. We are not funded by it. So whenever we come something like to look into, we'll use natural products. So while looking at Q13, we were using dose like 40 to 50 micromolars in TNBC cells. So we thought Q13 is inhibiting migration of TNBC cells, which is very important for cancer progression and metastasis. Can we bring this dose down by combining more phytochemicals to it? So this is a recent publication this year where we have combined Q13 with curcumin and berberine. And we found that this triple combination is really effective in inhibiting the ENT as well as the migration of TNBC cells. So here I'm just showing the IC50 of all the three compounds. So what we did is that we actually took this concentration like 1/10, so 10 times down, and combined the drugs and looked at the EMT markers. So here we looked at E-cadherin, N-cadherin, vimentin, beta-catenin, CD44, and MP9. So you can see that mesenchymal markers are going down, but you can also see that in MDM468, E-cadherin, which is actually a epithelial marker, is also going down. But that is the specialty of these cells. It has been shown that in 468 cells, E-cadherin expression is required for the migration and metastasis, because these cells collectively move. They are not like MDM331 that they move singly. So they move in sheet, they move in clumps. So if you overexpress E-cadherin in these cells, the metastatic rate is high. And if you reduce, you will find less metastasis. So E-cadherin is a driver of oncogenesis in these particular types of cells. As we can see that there is downregulation in the EMT markers, we next look into the migration, and we found that compared to individual treatment on the left hand side, the triple combination is more effective. We further looked into colony formation ability of 231 cells, and we found that triple combination significantly inhibit the colony formation ability of these metastatic MDM 231 cells. So if you look at the colony formation assay, what we do is actually you treat the cells with a drug and you allow the cells to grow and form a colony. Similar to like what we do in patients, we give the chemotherapy, and it is the residual disease that comes back and is the cause of relapse. So this residual disease, people are now saying it's because these are cancer stem cells, they are resistant to therapy, and basically when you look at colony formation ability, basically you indirectly looking at the cancer stemness. So we also thought if the CFU is decreasing, there is a population of cancer stem cells going down. So we looked at the CD44 and 24 population, CD4 positive and 24 negative, and we found that the triple combination is actually significantly inhibiting the stemness. Now, colony formation ability we have shown, we have now go one step up to show in 3D cultures that yes, this proliferation is inhibited, as you can see by the relative colony area on the right hand side in the graph. The triple combination is really effective compared to the individual combinations. We further show that because cisplatin resistance is a very common problem in TNBC patients, we further show that this combination, when given with cisplatin, is better than the cisplatin alone and the combination alone in both the cell lines. So this is our conclusion till now, that our data showed that elevated PFKP and HUR levels are associated with TNBC progression and relapse, and the ability of quercetin to inhibit these oncoproteins provide a rationale for using it as an anti-cancer agent. Furthermore, we put forward this viewpoint on the role of synergistic combination of these three phytochemicals, quercetin, curcumin, and berberine, and their ability to inhibit EMT process, migration, and the invasion of TNBC cells. So, further studies are warranted to validate the effect of this combination in vivo in TNBC cell models. So, my group is also working on gallbladder cancer and oral cancer, and we are taking this combination to there also. With this, I like to thank my Department of Medical Oncology, my funding sources, and my PhD students. So, on the topmost, it's Umar, who carried out PFKP work. Actually, he is working on beta-catenin mediated metabolism in TNBC cells, but I have given like a natural compound to everyone to look this aspect also. The second student is Arundhati. She looked at the HUR study, and the third one is Akansha. Actually, she is working on gallbladder cancer cells, but she finished this triple combination study this year. Thank you.
Thank you so much, Dr. Prasad. I think it was a very nice talk on the use of combination therapy for triple negative breast cancer. We can have one. Yeah, please.
Yeah, it's a good talk. So I have one question. When you are using combination of quercetin, curcumin, and DBR, how do you know that which one is entering at what concentration? So instead of that, because you have used mixture of all three, right?
Yes, sir. And individual also.
Right. That. But instead of using mixture of all three, if you can establish like some selective nanomaterial and chemically conjugate or encapsulate with these three compounds and show their stability and then drug release, druggable molecules.
Definitely, sir. It's a good question. Basically, we are
That
looking
will be
into
more
collaboration for
targeted.
that. Yeah.
More targeted, and second thing, you need to test in organotropic, orthotropic
Yes,
TNBC model.
yes, sir. That we are doing in triple negative breast cancer organoids, patient-derived organoids,
with a
and
selective
incorporate.
delivery
Yes.
mode of delivery. Thank you.
And my question is about your those glycolysis experiments.
Yes.
You checked the treated PFK1 you checked, right? The expression of PFK1 or PFK2?
Yeah, actually, I have not shown you the full data. Okay.
Okay.
So what we did is that we looked at We were actually looking at like non-canonical vent signaling.
My question is about, did you check the enzymes related with Krebs cycle also or not? Or
No, we only limited to glycolysis, where we looked at hexokinase 2 as well as PK.
So it is possible that
It
your
is
molecule
possible.
might be hitting to the
Yes,
enzymes
it is
related to
possible.
Krebs
It is
cycle also.
possible, and we have a DBT funded project where we are looking at beta-catenin mediated metabolism TNBC, where we are looking at both the components, aerobic characters as well as oxphos.
Thank you.